and thanks everybody for joining us today. I'm going to be showing you some of the new features that were released in LaserGene 15.2 a few weeks ago, and we'll cover some things in detail, and I certainly welcome any questions that you have regarding any of the materials that I covered today. So what's new in version 15.2? There are several new options in SeqBuilder Pro. The main feature is agarose gel simulation. We have a nice simulator that looks realistic and allows the user to configure the gel in many different ways. And so we'll spend quite a bit of time with the simulation. We also have some additional tools added to our batch sequence modifications. So this is powered by our SeqNinja application, and it provides some easy access to some of the powerful tools that are in SeqNinja. And so that's something that we introduced in version 15.1. We've added more in 15.2. We certainly would welcome feedback from our users as to any other tools that they'd like to see us add there. So we'll look at some of those as well. And then we have, and I have highlighted in green some things that we'll take a look at, but there are some other features. We have a motion library for proteins accessible through SeqBuilder. We also have a new help system called Manula that integrates tutorials with videos, and it's a really nice way to learn to use the software. So if you haven't looked at the Manula yet, certainly do that. And then in MegAlign Pro, we've added some pairwise alignments. These are some of the local and global alignments, Smith and Waterman and Needleman Wunsch, that you may be familiar with. But we are optimizing these with something called Parasail. And so we'll talk a little bit more about that later, so that they run faster and better take advantage of a modern computer and computing power. We've also added ClusLW, again, as we move to start to retire our classic MegAlign. We're moving that functionality over, in addition with some new functionality. And then some things that we aren't going to cover today, I'll just mention some enhancements for our RNA-seq workflows, where we utilize DE-seq2 from Bioconductor. So this accesses that from the cloud. And looking forward a bit, we have improved some of our cloud assembly software. So it is cheaper, faster. It's essentially a beta now. We have some customers trying this out. I would imagine our next release will have a full-fledged release there with cloud functionality. So just a picture, again, reminder, we're going to go through gel simulation in some detail here, where we get some nice images. We also have these MegAlign Pro pairwise alignments, so a whole new view is built in MegAlign, lots of flexibility as to how we can do pairwise alignments, and we can do them from our multiple sequence alignments. So we'll take an in-depth look at both of these pieces of functionality. And then I did mention Parasail. So this is a library that we're using, and it really optimizes and it allows these older algorithms to use modern CPUs, right? So you get a lot more performance then from these older algorithms, and we'll be looking at some global and local, and show a couple different data sets where they're applicable. And one more slide here, and then also this is kind of a first for DNASTAR. We are starting to utilize some of the open source and powerful tools up on Bioconductor, and we try to do it in a way that it's seamless to the user, and you don't have to know how to run and install some local R package, but we integrate it in with our software and make it seamless for you. And we'll have some tutorials and videos that are specific for this entire workflow. So DE-seq2 is the first. We'll have more in the future, and so that's something that we will look to expand. So with that, I'll jump out of the PowerPoint, and we'll start with SeqBuilder Pro. And so SeqBuilder Pro, if I'm going to do a gel simulation, there's actually multiple different ways to start the simulation. I'll start here just by a new agarose gel, and it brings up a new SeqBuilder Pro window. Depending on the gamma that's on your screen, You may or may not be able to see the kind of the gel lanes here. You'll see the numbers 1 through 8, and then there's a size marker on the side. And when you're looking at this through your own computer, you'll see very faintly some of the gel lanes. And this gel then has a couple different views. There's the simulation view at the top, and then a fragment size summary view at the bottom. And one of the nice aspects to this is that if I look over here on the right in the settings, there is a number of different settings that I can change to make this gel look like the gels that I'm using. And so I can change, for instance, the brand. You know, I can switch to a Biorad gel or you know, an Owl gel box. And then we have the types entered in, the typical types of gels that we would have. And you notice that as I change the options, the image changes as well. And I can set the number of lanes. Here's 10 lanes, there's 40 lanes. I can also add another comb, you know, so it really gives us lots of flexibility then to make this gel look or like the gel that we're using. I can also directly, you know, put a custom gel in essentially, and I can type in, you know, here I have a 14 centimeter length gel, and maybe my gel is more square, and then I can just change that number and it will update the image. So high degree of flexibility in making this image, you know, look how we need it to look. And I can change that back. So that's adjusting this image. And I can also change the agarose percentage, and we'll put some markers on in a bit, and I can show you what that does, as well as some other. So let's add a marker here. So if I go and switch the tab to markers, and I'll stick with Pro Mega Marker. There's a couple different ways that I can start loading the gel now. One way is to select the lane and then use the checkbox to pick the marker. I can also drag and drop, and so I can select something here and drag the marker into the view. So again, depending on what you're comfortable with and how you like to, we had some, when we showed this to the first users, the Mac users wanted to, you know, they tried to drag and drop, and we realized, well, we really need to add drag and drop support throughout this. And so whereas the Windows users were more likely to check a checkbox. And so there's different ways then that now, so we can change this. You can see that I can select the lane, I can delete, I just right click, delete if I don't like something that I loaded. So I can copy and paste, so you can edit this as well. So I may put a gel here or a marker here and say, well, I don't want it there, I actually wanted it in this lane. You know, so I can move things around, it's quite flexible. And then of course, once I've put some markers on, I can see again the simulation of the runtime. There's a little slider here, so I can go to time 1 minute and progress then. And what's nice about the simulation is I can see, well, at that time and that gel percent, do I resolve the size fragments that I need to resolve, or do I need to really change my gel percentage a bit? You know, so maybe I go to a 1.25% gel. The artistic options here, blur, smile, those are added. I could just have this more of a simulation with just white lines, or I can make it look a little bit more like a gel by adding those artistic effects. So that's how you change the size of the gel, the number of lanes of combs, the agarose percent. Now, in terms of loading sequences on, of course, there's more than one way to do it, but I'll show you one of the ways is to go to the Sequences tab here. So I'm in this upper right panel, and I can import sequences. And so I've got a few here on this computer. So I'm going to take these 4. And these are larger sequences. One of them is a 250,000 base pair chunk of the E. coli genome. So we're going to load some larger fragments here. So now I have this fragment list. They aren't applied to the gel yet. One way to load a lane is to select the lane and then pick the sequence I want in that lane. So it could be any order. So there's a lane CG, and now I'm just selecting a lane. And I can also drag and drop from this area, right? So I can move them that way. Again, I can cut, paste. So again, edit the gel as I see fit. Okay, so there's some uncut sequences loaded onto the gel. Now I can apply enzymes to cut these. I might also want to look at a particular sequence. So if I want to look at, let's see, the K12NG1655, the fragment of the E. coli genome, I'm just going to double click here. And it's a big sequence, so it'll take just a little bit to pop it open. Oops. There we go. So this is, again, a floating window. So there's that sequence. So you might work on a sequence this way. And I can see I've got some echo, Hindi, and I think, yeah, and a BAM enzyme applied to this sequence. So maybe that's what I'm interested in, you know, if I'm trying to cut out a certain piece or digest in a certain way. And so to apply those same enzymes that I have in my map to this sequence, I can go to the Enzymes tab, and there's a smart filter here. So I can type in echo and apply it. And so now we've digested this large sequence with echo R1. If I want to add another, I'm going to hold my shift key down and click. And I'm just typing my keyboard to bring, and so I can apply those enzymes. Let's go back to echo, hold shift down. And so that almost looks, you know, it's digested then with 3 different enzymes. I can zoom. When I zoom in far enough, you can see at the top of the gel here, it gives me a change in the icon, so I can see that this fragment has been, this DNA has been cut. It lists the enzymes. There's a pop-up window. Also the fragments. There's a list now that shows this fragment of the genome cut with these 3 enzymes, and gives me a list of those fragments. So I can zoom out again here, and it is interactive. So this is, if I select a fragment on the gel, I get that fragment selected. It's red here in my fragment list. I also get that piece selected then in my sequence because I have that sequence open. So if I select here at the top, that's gonna be a much larger wedge. So there I've got a large fragment, a large selection here selected. So there's, again, multiple different ways that I can apply things. Okay, so let's go, so I'll go over here to the, there we go, okay, reset. So I'll refocus on this other sequence. Again, go back to another enzyme. And I can get, if I have shared fragments, I can get a different color on the enzymes or on the bands, if they are all contained in another one. And oops. I can also change the view if I want to make it, there's a little icon at the bottom that is a splitter. So if I don't want to, I can either resize this way or click the icon to give maximal real estate to a gel. And then of course I can edit and bring in new fragments. So I'll bring in a different type of sequence now. So these are large sequences. I can also import a different type of a sequence. So I'm going to clear out one that has PCR products. So we'll make this gel look a little bit more like a PCR gel. So let's go to something with more lanes on it. Let's see if I can do a 16 lane gel, and we'll change the marker, and we'll change the percent, and I'm going to try some different kinds of markers now. So we'll have some standard from Promega. So we've got a 25 base pair ladder, or 50, 25, 10. And so this is kind of a way to try out markers too, and say, well, here's some markers that I may buy from a particular vendor. Which markers are going to be most suitable for my needs? So I can load them up on a gel. And I'm going to bring in a sequence now. Actually, I'll open the sequence up. So I'm going to close this one. And I'm going to open a sequence that has some primers that were designed. So so this is again about a 10 kb chunk of E. coli, and there's several pairs of primers. So my primer list here has 4 sets of primers, and again these were designed by selecting a CVS and doing a primer, create primer pairs. So when primer pairs are created in SeqBuilder, they automatically create a set of features, and those features contain both the primers and the product, and they're saved within the file. And so I can do this a different way now. I'm going to close my gel. Actually, so if I'm working on this sequence, I can now say I want to run this on a gel, and so right now it loaded that sequence, but I can just clear that out and go to the primers tab. And the primers tab then identifies that I have the 4 pairs here, and so I can load in, and this is essentially just putting in the PCR fragment from that sequence. And now I can again set markers. So if I've decided what I want for markers, I could have the PCR marker. I'll make this give more real estate here. And again, I can try some others just to see which ones resolve the PCR fragments that I'm interested in. So there's a set of markers, or I could have a list. It could be primer sequences that are in a FASTA file. I could bring all those product sequences in and load those onto the gel. So there's multiple different ways that I can do that. We'll go back to the settings here, and you can see at a 1% gel, we get things that kind of don't resolve very well, and I can change this, change it to a 2%, or change to a 3% gel, again, depending on the size, and resolve, do the runtime, and be able to pick the right size markers, for the size. I also have a reticle at the mouse option. So if I need to size things, this can help me a little bit. And you'll notice in the bottom left corner, it tells me exactly what base pair position that's at. So all in all, a very useful tool. It allows me to set custom sizes on my gel, dimensions that you'd get from the common commercial brands, or custom dimensions. All the different combs and number of lanes are supported for those gel brands. I can have 1 to, I think, up to 4 combs, so I can make a very large, complicated gel if I'd like to. Different ways to bring sequences in. I can open a sequence document, launch a gel, and it'll load that data in. I can drag and drop sequences and or import them. So highly flexible, makes a nice image. So it's a nice new tool. So that's gel agarose simulation. I'm going to go on now to some of the new batch tools that we have. So I'll close these out. And so under our tools menu, again, this is some new functionality that was introduced with version 15.1, where we have doing things in batch. And again, this is powered by SeqNinja. So we have a tool that is has less GUI interaction than it does through SeqBuilder Pro. And so we thought that a lot of those tools would be useful in the context of SeqBuilder. So we started to move some of them over. And you can see some new ones under sample by. So this allows me to go through a set of sequences and collect subsets from that set using different criteria. And so one thing is to sample by length is something that's fairly common. So I'm going to let's do a sample by length, and let's find my sequences here that I want to use. I can. So here are some sequences that are some RefSeq mRNA sequences. And these might be something that I've downloaded or that I've translated or de novo assembled using our software. So 410, so I'll pull those in. It'll take just a few seconds to import them. And so now we have a set of sequences imported in, and I can select, and you can see that if I can select the whole folder, individual sequences, and then I can sample them. So with mRNA, sometimes there's fragments of mRNA and I want to size select. So a common thing would be to set a minimum length of some distance. All right, and I can use a number stepper or a range of sequences. And when I click run, then it'll just go through all the sequences and sort out and collect only those that are at least 1,000 base pairs long. So the result folder then shows all these sequences that are 1,000 base pairs long. And we can collapse this so you can see here's the output sequences. Now I can do a further action. For example, I may want to do a different kind of a sampling. So I can switch the tool and I could sample by end tags as well. So what this is going to do is, again, I can select this group. There's 373. I may want only the subset that starts with ATG. Now these are annotated, so it's not as necessary, but so I can look for tags at the start or ends of the sequences. All right, and then so I get a little subset here. I could create a folder to put them in. And from here, you could do, and I'm just kind of showing you, I could do things, if I've got a process here that I need to do, it could be translate, so then I could translate those. So I'm just stringing together a couple of functionalities. And you can see that with the translate, if I've got FASTA files that do not have, where the files do not start at ATG, for example, and I just want to find those sequences that have started ATG, I can do that, or I can have the translator look at the annotation and translate from the different as annotated or from specific frames. pick a genetic code and the file output, and again run, and it will run the translations. So it's a nice way to do batch operations, and I know folks are reverse complement, something as simple as that. If it's one sequence, not a problem, but if you've got a few thousand that need to be reverse complemented, it's nice to have a tool where you can dump the sequences in, run the tool, and get the output. So again, these are a tool set that we've continuously been adding to. There are other sample buys. We have things like probability, so I can run. Here, I'll remove some of these now so we don't have too many built up. So I can run and sample by every sequence, or do a random collection of sequences. So there's a number of different ways that we can, go through a larger set then and create a subset. And so, here's a random sample containing, 10 sequences. Like that. So useful tools. We will be looking for, customers are giving us some feedback as well. And so, if there are tools like this that perform batch operations that you're looking for, that you have a need for, please, feel free to pass that information on to your sales rep, or you could chat it in today. And, we're looking for that kind of feedback to there's a lot of tools we could add, but we'd like to pick the ones that people are most interested in and add those next. So that is SeqBuilder Pro. From here, I'm going to go over to MegAlign Pro. We're going to look at some of the pairwise alignments. And so to start here, I'm going to add some sequences, and we'll do a multiple sequence alignment first. So I've got some, let's see, HPV genes here. Let's see which one. So I think these are whole viral genomes. So we got some viral genomes, and we will align these with, we'll start by doing a cluster omega alignment. I'll take just a bit here. So a common workflow is, do alignments of multiple sequence alignments of a set, and then identify individuals in that set that are more closely related, and then do a different type of an alignment. Do a pairwise alignment on individuals that are either close or more divergent from each other. And so we have some new views now in MegAlign Pro that facilitate that kind of a workflow. And we'll let this finish up. Should just take a few more seconds here. And again, if you have questions, I'm moving through quite a bit of different material. At the end, we'll finish up here after the pairwise, and I will be around to answer any questions that do come through. So I'll make this a little bit larger. So we have a pairwise alignment, and the way MegAlign Pro works, of course, is we have a different set of views. There's a tree view, a distance matrix, and now we have a sequences view, and then of course a new pairwise view. And I'm just going to reorganize this. I'm going to move the tree to the top, and then as well as the sequences view. And so I can customize this view to be, how I want it. And I'm just going to resize a little bit. Okay, so this is more along the, so I can do analysis either from the tree and find, you can see this is a pretty good set of viral genomes, and I can identify individuals that I may want to do a pairwise alignment on, either based from the tree, or it could be from, I may have individuals here in sequences view. And so I can select sequences, and then I can go and do a pairwise alignment. And you can see the Pairwise has a whole set of different options now. And so I can align using local Smith-Waterman, a global or a semi-global Needleman-Wunsch. There's a few alignment parameters here. I won't go into detail here, but there is help that helps describe what these are. And so this kind of brings us to another new feature of the software, and that is it's called a Manula help, which is a new way to provide help to our customers. And you can see that on the left side is kind of a tree overview of different things that I can collapse down. Right, and then here is, and then each individual topic. Some of them have tutorial data that goes with. There are videos that are embedded in here. So it's really a nice way to help learn to use the software and learn more information about the features in the software. So, and you'll see this now in MegAlign Pro and also SeqBuilder Pro. Eventually, all the DNASTAR applications will have this new format of help, which is really tremendous. So I can set, so these icons then, help icons lead us to contextual help on those specific topics. You may or may not set parameters, and now I have a pairwise alignment from sequences that I selected in a multiple sequence alignment. And I can give it more real estate by clicking this icon in the lower right corner. And we get a little, you can see in the header here, it tells me what the target sequence was, HPV10, and the query sequence, HPV11. And from the pull down now, I can do more pairwise alignments. So this is one pairwise alignment, and I see in the header, it's a local Smith-Waterman, and I see the gap penalties and the matrix that was used and identities. So we get the kind of the summary of the alignment, and then I can see the actual alignment. Now, if I want to do more alignments, I can actually make multiple pairwise views. And so I've collapsed everything down, but there are some quick buttons here. There are change the alignment options. I can create a new alignment from a selection, or I can say I'm going to have multiple pairwise. So now I have pairwise 1. Here's pairwise 2. From the pull down, I can change it to, and here's a third one, and I can select new sequences. So now I've got 1, 2, 3 pairwise alignments, and I can keep adding them, and they'll stack up, and I can have, you know, as many pairwise alignments of the sequences that have been loaded into the project as I want. I can also go back and change, you know, parameters. So this was the 2 sequences with a local Smith-Waterman. Maybe I want to redo this in a semi-global, right? So I can quickly change a parameter or use a different algorithm to change the alignment. Now there are also, I'm gonna move this controller over here. There's also under the style panels, we've got some new things specific for pairwise alignment. So I can do things like just make it linear, depending on your preference. I can, you know, as you would expect, I've got lots of flexibility now with what's highlighted, color only things that are different or similar, color the background. I can change the sequence, you know, so that it's solid. All right, and then change foreground. So just showing you some options here to, you can make this look however you want it. So I can have a very different looking pairwise alignment or not. So it's really a nice way to present pairwise alignments. Of course, I can export these out. So there's let's see, right here, a little camera, export the image out, and I have options PDF documents or PowerPoint documents or bitmap images. So real nice way to get the images out of the software. So that's one type of alignment. Let's do, we've got a couple minutes here, so I'm going to show one other type that is for, in this example, it will be aligning small sequences to a big sequence. So in this case, CDSs to a bacterial genome. And so I'm just going to move this aside here and load the sequences in. And so that's under, this is some E. coli, and here's some sequences. And again, we're putting a genome in, so it'll just take a few seconds to load the genome in. Okay, and we can see in this overview at the top is kind of the sanity check, and it just tells me there's one really large sequence here and several very small sequences. And as you know, it's easy to mistakenly sometimes add a genomic sequence. And so this overview just kind of tells you before you run the alignment what the relative size is. And you can see in this case, if I zoom way in, I've got some relatively small sequences and a big sequence. And now in terms of doing pairwise, I can go to the Pairwise View directly, and I can change my target. I can say this is, my target is the genome, and I'm going to align this sequence. So it's one of the genes. All right, now if I want to do the other, I can clone the view a couple times. And so now I'll have a view for each one of those. Each one of the CDSs now is, and you can see the speed here. We're aligning this to a genome. It's going fast. And that again is part of the Parasail optimization. It allows me to do these alignments pretty quickly. Even though we're using, an algorithm that's a few decades old. I think it's from the 1980s. I think it's Smith-Waterman. And again, I get some information about the alignment. I can edit this. Lots of flexibility in how I can make this image look in the software. So with that, I think we've, again, thanks for your time, and I was happy to be able to show you some of these new features in version 15.2, and I'd be more than happy to answer any questions that have come through.
Thanks, Matt. This is Sharon again, the organizer. I've gotten some questions from our participants while you were giving your webinar, and I was able to answer most of them using the questions and chat function, but I saved a couple of them for you.
Thank you.
So the first one I'll start with, since you have MegAlign Pro open, I'll do this one. What is the capacity for pairwise alignments in MegAlign Pro?
So pairwise alignments, it really depends on the alignment algorithm that you're using. If it's a local alignment like this, you can align a short sequence, a gene or CDS size sequence to a chromosome or a whole genome. If it is a global alignment where it's two sequences that are about the same size, the size limit is about eukaryotic gene size, so 50 to 80,000 base pairs in size. So it's always important to know, what it is that you're trying to align and make sure that you're selecting, the right algorithm to do that.
Thanks, Matt. So for the next question, you're going to have to get SeqBuilder Pro back up. And that question is, can I create custom molecular weight markers? I think that's regarding the agarose gel simulation.
Yep, I can do that. So I will. Okay, so let's go back to one column. Okay, so if you go to the markers tab, there is at the bottom left corner here some little toolbar options. So I can add a marker. And let's see. And then you can type in sizes. So 10,000. So this would just be a custom, oop, I don't want 75,000, so let's go 75. Let me try that again here. Okay, 10, 1, 2, 3. So it's a simple tool, not terribly sophisticated, but allows me, in the case that I have a marker that is not in our list, to add one. So there is my markers, and you can see that at the very end it just says Matt's markers, and I will load Matt's markers in, and there we go. So it's these little toolbars at the bottom allow me to make custom markers.
Great.